Hello students, welcome to the EPG Pathshala. I am Dr. Rajeshwari Sinha, working as Program Officer at the Center for Science and Environment. In this lecture, today we are going to learn about DNA structure. In this lecture, we will have the following objectives: to understand the nomenclature of nucleic acid deoxyribonucleic acid or DNA as a genetic material, to understand the structural characteristic of DNA with respect to its replication and genetic code, to understand the base composition and sequence in DNA. We will also see the key features of the DNA structure and also the various structural types of DNA. Let us have a look at the objectives that will be covered in this lecture. We will understand the nomenclature of nucleic acids, look at DNA as a genetic material, understand the structural characteristic of DNA with respect to its replication and genetic code, understand base composition and sequence in DNA, the key features of DNA structure, and also know about the structural types of DNA. Nucleic acids are polymers of nucleoside and monophosphates, which are called nucleotides. The monomeric units consists of a nitrogenous base, which may be a purine or a pyrimidine, a pentose sugar in furanose ring form, which can be ribose or deoxyribose, a phosphate group esterified to the sugar. The coupling of base to sugar yields a nucleoside, which becomes a nucleotide when it is phosphorylated. Nucleotides can be linked together by phosphodiester bonds between the 3-hydroxyl on the sugar of one nucleotide and the 5-phosphate on the sugar of another nucleotide. Figure 1.1 illustrates the structure of such a dinucleotide. The addition of another nucleotide at the 3-hydroxyl end will produce a trinucleotide, and with many such nucleotides added, eventually a polynucleotide will form. Note that these compounds have polarity. One end of the molecule is different from the other in the sense that the pyrimidine nucleotide in the figure has a free 3-hydroxyl group on its sugar moiety, whereas the purine nucleotide does not. In the long chain forms nucleotides in this manner, one speaks of the 5' end of the molecule and of the 3' that is free hydroxyl 3' end of the molecule. The dinucleotide illustrated in figure 1.1 can be represented as an abbreviated form as phosphate G, phosphate C or phosphate GC, which show that there is a phosphate group at the 5' end. The 5' nucleotide is a guanidine. There is no phosphate on the 3' end. Note the convention of writing nucleic acid structures with the 5' end on the left. Polynucleotides containing ribose as opposed to deoxyribose are called polyribonucleotides. Naturally occurring polyribonucleotides are also known as RNA or ribonucleic acid. With few exceptions, the bases found in RNA are guanine, cytosine, adenine and uracil. If the dinucleotide illustrated in this figure was modified in the 2' position of each sugar moiety by replacing the OH by H to give a deoxyribose, we would have a deoxyribonucleic acid or DNA. In this slide, the structures of the 5 common bases found in nucleic acid has been shown. The basic structures of the bases are purine and pyrimidine. The 5 bases are adenine, guanine, cytosine, uracil and thymine. The 4 bases which are present in deoxyribonucleic acid or DNA are adenine, guanine, cytosine and thymine. The 4 bases which are present in the ribonucleic acid or RNA are adenine, guanine, cytosine and uracil. DNA as the genetic material. Genes of the eukaryotic organisms are made up of proteins and nucleic acids. For a long time, it was thought that the protein moiety carried the genetic information and that the role of nucleic acid was only a structural one. However, the DNA now is known to be the carrier of genetic information in all organisms except certain viruses which use RNA for this purpose. Early work with pneumococci. The first solid evidence came in 1944 from the work of scientists Avery, MacLeod and McCarthy on the transforming principle from pneumococci. Earlier in 1928, Griffith had found that non-pathogenic mutant pneumococci called R forms because the mutant strain could not form a capsular coat and thus appeared rough on the plates, could be transformed into the pathogenic forms called S forms because the capsulated organisms appeared shiny on the plates. The transformation was accomplished by injecting mice with a mixture of heat-killed S form and live R form pneumococci. The live R form was the non-pathogenic form. The mice died and the live S forms pneumococci were recovered from their blood. When either live R form or heat-killed S form pneumococci alone were injected, the mice survived. Avery et al. Purified extracts made from heat-killed pneumococci and showed unequivocally that the transforming factor was a deoxyribonucleic acid. Studies of bacteriophage T2, a virus that infects the bacterium E. coli, supported the conclusion that DNA was a genetic material. The T2 phage consists of a DNA core surrounded by a protein coat. In 1951, Heriot suggested that the phage might function like a hypodermic syringe injecting a nucleic acid into the cell with the protein coat remaining outside. The idea was tested by Hershey and Chase, who labeled T2 phage with radioactive phosphorus in the nucleic acid moiety and with sulfur in the protein. After infecting E. coli cells with the labeled phage, they were able to show that the radioactive phosphorus remained with the cells, whereas the radioactive sulfur was largely lost. Basic structure of DNA. Using data from X-ray diffraction studies by Franklin and Wilkins, Watson and Crick in 1953 proposed a structure for DNA that has become known as the double helical structure. This structure, which since has been amply confirmed, allowed Watson and Crick to propose a mechanism for the replication of DNA that in all essentials still is believed to be accurate today. Two antiparallel polydeoxyribonucleotide chains are wound around each other with the purine and pyrimidine bases on the inside of the helix and the deoxyribose and the phosphates lying on the outside of the helix. The planes of the bases are perpendicular to the axis of the helix, allowing stacking of the hydrophobic rings. Hydrogen bonding occurs between pairs of bases, one base on one strand pairing with another base on the antiparallel strand. Adenine, symbolically represented as A, always pairs with thymine T, and guanine, represented as G, always pairs with cytosine C. The above explains Chargaff's rules, which state that in nearly all DNA studied, the content of G equals the content of C. The helix is 20 Armstrong in diameter. In crystalline DNA, the bases are separated by 3.4 Armstrong along the helix axis, and each base is rotated 36 degrees in relation to the previous base. This means that in this type of DNA, the helical structure repeats at intervals of 34 Armstrong, that is every 10 base pairs. In solution, the helical structure of DNA probably does not repeat itself after every 10 base pairs, but nearly after every 11 base pairs, with the bases inclined 20 degrees from the horizontal. Genetic information is stored as the sequence of bases along the chain. Diassociation and reassociation of DNA's double helical chains. Double helix duplex chain of the DNA have a remarkable ability to dissociate from one another and reassociate once again. This behavior is essential to the process of replication and transcription. Denaturation, the rupture of the hydrogen bonds between the bases resulting from increasing temperature or alteration of the hydrogen ion concentration causes the two strands to come apart. Increasing pH deprotonates ring nitrogens of guanine and thymine. Decreasing pH protonates ring nitrogens of adenine, guanine and cytosine. Because increasing acidity can cause the rupture of purine glycosidic bond, temperature change is the most practical way to denature DNA for reassociation studies. The dissociation of the two chains with increasing temperature occurs at the definite temperature, and thus the process is referred to as the melting point Tm. It is the temperature at which 50% of the double helix has been unwound. The melting of the DNA can be studied by measuring the absorption of light at 260 nanometers. Melting causes unstacking of the base pairs, which results in increasing absorbance, also known as the hyperchromic effect. The melting temperature is strongly influenced by the base composition of the DNA. The DNA rich in GC pair has a higher melting temperature Tm than DNA which has a higher proportion of AT pairs. The Tm of DNA extracted from different species and measured at pH 7 in an isotonic salt solution varies linearly with the GC content, with synthetic poly AT having a Tm of about 65 degrees Celsius and a synthetic poly GC having a Tm of about 105 degrees Celsius. Renaturation or annealing. If the temperature of the melted or dissociated duplex DNA is rapidly reduced, the original double helical structure does not reform. If, however, the temperature is held at a value of about 20 to 25 degrees below the Tm, the original double helical structure will reform. The rate at which the structure reforms or anneals is related to the concentration of complementary sequence. A measure of the complexity of duplex DNA genomes can be obtained from annealing studies in which the fraction f of single-stranded DNA remaining is plotted against the logarithm of the product of the initial concentration CO of a single-stranded DNA in moles per liter times T in seconds. The plots of f against log COT form a sigmoidal curve. The genome of the phage T4 reanneals more rapidly than the larger genome of E. coli. The very much larger non-repetitive fraction of calf thymus reanneals about 3 orders of magnitude more slowly than that of the E. coli. Eukaryotic DNA contains many sequences of DNA that reanneal very rapidly. The size of the DNA molecules. A very simple chromosome such as that of the SV40 virus has only about 5 to 6 genes, 5 to 43 base pairs, and is about 1.7 micrometers in length. More complex organisms require more DNA, and thus the genome of E. coli is more than 1 millimeter long and contains 4 into 10 to the power 6 base pairs. Measurement of the very long DNA is difficult because of the tendency of the molecules to shear, but stretches of DNA about 1 millimeter long have been measured. Human chromosomes with molecular weights up to 160 into 10 to the power 9 Daltons corresponding to 240 into 10 to the power 6 base pairs would, if continuous, measure up to 8.2 centimeter. Studies of the viscoelastic properties of the DNA have revealed the lengths of different DNA molecules in the chromosomes of many cells, and there is a very good agreement between the length of the DNA in Drosophila chromosomes as measured by this technique and the DNA mass in Drosophila, which has been determined cytologically. DNA molecules run the full length of a chromosome without discontinuity at the centromere, and the continuity is conserved throughout the cell cycle. These data provide the most compelling evidence that a chromosome of a higher organism is composed of a single molecule of DNA. The shape of DNA molecules. Chromosomal DNA may be linear or circular. DNA molecules do not exist in an extended form in either case. Rather, both rods and twisted or supercoiled into complexes that result in a several thousand-fold condensation of the DNA in a chromosome or the head of a virus. In eukaryotic chromosome, DNA is complexed with histones which profoundly influence the structure of the chromosome. Non-histone proteins and possibly RNA also may affect the packaging of DNA. When a single molecule of DNA is complexed with histones, it forms a chromatin thread, which is a beaded string coiled upon itself. In DNA viruses, DNA condensation is achieved by regular spool-like wings assisted by polyamines and metallions. Supercoiling of DNA is essential for stages of replication, transcription and recombination. It occurs only when there is a restraint upon the rotation of the end of the DNA chains, such as which occurs either in circular DNA or in linear DNA when the ends are too far apart to allow rapid spinning. Most naturally occurring DNAs are supercoiled or supertwisted. The degree of supertwisting influences the degree of unwinding of the double helix, with the supertwisted DNA containing more base pair per unit length than are normally found in the double helix. Circular DNA relieves this torsional strain by twisting in the opposite or negative sense. Supercoiled DNA relax under the following circumstances: if one strand is nicked followed by resealing, if certain chemical agents such as ethidium bromide are inserted, by binding to specific unwinding proteins prior to the process of replication. The degree of supercoiling, both in terms of its generation and its removal, can be measured by the following techniques: electron microscopy, sedimentation of DNA complex with ethidium bromide, agarose gel electrophoresis, study of one of several enzymatic functions that depend upon supertwisting, some prokaryotic enzymes which act on negatively supertwisted DNA, eukaryotic enzymes which act on both negatively and positively supertwisted DNA. We have now reached midway into this lecture. So far, we have looked at the basics of the DNA structure and how the basic units of DNA, which is the pyridine and pyrimidine bases, are organized into the DNA along with the sugar and phosphate moiety. We have also looked at the important historical experiments which have led to the establishment of DNA as a genetic material. We have understood the concepts of denaturation, renaturation and annealing with respect to a DNA molecule. Now, let us move to the next section. In the next few slides, we are going to look at some of the key features of the DNA structure, some different structural types of DNA and their role, function and importance. Key features of the DNA structure. DNA is a right-handed double helix. The bases in opposite strands hydrogen bonds according to the AT and GC rule. The two strands are anti-parallel and run in 5' to 3' directionality. There are approximately 10 nucleotides in each strand per complete 360-degree turn of the helix. Nucleotides are linked by phosphodiester linkage. It has been estimated that half-life for hydrolysis of phosphodiester linkage in DNA is about 200 million years. Types of DNA on the basis of structure. aDNA structure is shorter and thicker, found under conditions of low humidity and high salt concentration. There are about 11 base pairs per helical turn with vertical rise of 2.56 angstrom per residue. there exists very deep major grooves and shallow minor grooves. Displacement of the axis also forms a hole approximately 3 angstrom in diameter. The bDNA. The average overall structure of DNA in living organisms is believed to be the bDNA-like. Notably, this conformation, unlike the A and Z form, is highly flexible. This is found under the conditions of high humidity and low salt concentration and was the basis of the Watson-Crick structure. The helix is relatively long and thin with approximately 10 base pairs per helical turn. The rise per residues is 3.4 angstrom with approximate thickness of bases. The base pairs are nearly perpendicular to the helical axis. The major and minor grooves are roughly the same. The zDNA. It incorporates a left-handed helix rather than the usual right-handed variety of zigzagging backbone. The zDNA structure is longer and much thinner than that of bDNA and completes one turn in 12 base pairs. Major grooves are not distinct and minor grooves are very deep. The biological functions of zDNA is not known. Some evidences suggest that zDNA influences gene expression and regulation. The bent DNA. DNA sequences with runs of 4 to 6 adenine faced by 10 base pair spacers produces the bent conformation. DNA bending appears to be a fundamental element in interaction between DNA sequences and proteins that catalyze central processes such as replication, transcription and site-specific recombination. Bending also occurs because of photochemical damage or mispairing of bases and serves as a recognition signal for initiation of DNA repair. The cruciform DNA. Double hairpins, often called cruciform structures, can be found in some DNA sequences. A special kind of sequences referred as a palindrome is required. The word palindrome is of literacy origin and usually refers to a statement that reads the same backward and forward. The overall biological function for cruciform is only circumstantial and has not been established. Triple-stranded DNA or the HDNA. The normal Watson-Crick pairing is AT and GC, but later it was discovered that deoxy triplets like TAT and C plus GC can also be formed, which is known as Hoogsteen type base pairing. HDNA may play a role in the control of transcription, replication and recombination. 4-stranded DNA. Guanine nucleotides and highly G-rich nucleotides from novel tetramer structures called G-quartets. 4-stranded structures are stabilized by the presence of metal cations, especially sodium and potassium. X-ray diffraction and NMR experiments have shown that C-rich oligodeoxyribonucleotide can form a complementary novel type of 4-stranded complex called the iDNA. Importance of 4-stranded DNA. The telomerase activity has been detected in most tumor cell lines and may be responsible in cancer. A new approach towards cancer treatment is emerging as telomerase inhibition, which involves use of the drug that binds with the G quadruplex DNA. Large aromatic molecules such as porphyrins and anthraquinones selectively bind and stabilize G quadruplex DNA structure. Slipped DNA. DNA regions with direct repeats symmetry can form structures known as slipped or mispaired DNA, often abbreviated as SMP DNA. Although SMP DNA has not yet been identified in vivo, genetic evidences suggest that this type of DNA has undoubtedly involved in spontaneous frame shift mutagenesis. Duplication of certain simple triplet repeats that are implicated as the basis of several human genetic diseases may also occur by this mechanism, like the Fragile X syndrome. Students, we have now come to the end of this lecture. In the previous section, we looked at the various types of DNA structure, for example, the aDNA, bDNA, the zDNA, the bent DNA, the cruciform DNA, the triple-stranded and 4-stranded DNA as well as the slipped DNA. we understood what are their properties and how these are different and unique from each other. Now, let us summarize once again what we have learnt in this lecture. A striking characteristic of DNA is its ability to encode an enormous quantity of biological information. This information is stored in the nucleus of the cell. The ability to store large amount of information on molecules and to access it readily is still far beyond modern information technology. The capacity of nucleic acid to maintain and transmit this archived information efficiently arises directly from their chemical structure. Thus, DNA seems to be not only and self-replicating, but also in the knowledge of recent development may become self-analyzing and problem-solving ultimate mechanism in future. Thank you for watching this lecture on DNA structure.